Hello everyone, and welcome to today's session. I'm excited to introduce you to Module 3, Enzymology. In this presentation, we will focus on Study Session 2, which explores the structures of DNA and RNA. This is part of BCHM 241, our course on the biochemistry of macromolecules. Today, we'll delve into the fundamental structures of these vital nucleic acids, understand their differences, and see how their structures relate to their functions. Let's get started and explore these fascinating molecules together. Today, I want to walk you through the structure of DNA, which is fundamental to understanding how genetic information is stored and transmitted. We'll look at how DNA duplicates itself during cell division and how RNA is synthesized as a key step in gene expression. It's also important to recognize the role of some synthetic nucleotides, which are crucial in various biotechnological applications and research. These synthetic building blocks help us understand and manipulate genetic material more effectively. As we explore these concepts, keep in mind how they connect to the bigger picture of molecular biology, emphasizing the precision and complexity of genetic processes. Understanding DNA structure and function is essential for grasping how life operates at the molecular level. In this slide, we're summarizing the key points from M3SS2. First, we want to highlight the main concepts we've covered so far, which include understanding the core principles and their practical applications. It's important to see how these ideas connect back to our overall theme of building a strong foundation in this subject. By reviewing these highlights, we reinforce the critical aspects that will support us as we move forward. Remember, the goal is to not only grasp the individual points, but also to see how they fit together to form a comprehensive understanding. This summary acts as a bridge, tying together the concepts we've discussed and preparing us for what's next. Let's take a closer look at the structure of DNA. DNA, or deoxyribonucleic acid, is the molecule that carries the genetic instructions for life. Its structure is often described as a double helix, which looks like a twisted ladder. The sides of this ladder are made up of sugar and phosphate molecules, forming the backbone. The rungs of the ladder are composed of pairs of nitrogenous bases. These bases are paired specifically. Adenine always pairs with thymine, and cytosine pairs with guanine. This pairing is crucial because it ensures the DNA can be copied accurately during cell division. The way these bases are arranged encodes the genetic information. Understanding this structure helps us grasp how genetic information is stored, replicated, and passed on from one generation to the next. It's fundamental to everything we'll explore about genetics and heredity throughout this presentation. Now, let's talk about complementary base pairing. This is a fundamental concept in understanding how DNA maintains its structure and replicates itself. Essentially, each base on one strand pairs with a specific, complementary base on the other strand. For example, adenine always pairs with thymine, and guanine always pairs with cytosine. This pairing is due to the specific hydrogen bonds that form between these bases, ensuring the stability and accuracy of the DNA double helix. Recognizing these pairing rules helps us understand how genetic information is faithfully copied during cell division. It's a key piece of the puzzle when exploring DNA's role in inheritance and molecular biology as a whole. Let's take a closer look at the double helical structure. This is really the iconic shape that DNA adopts, and it's fundamental to how genetic information is stored and replicated. The double helix consists of two strands that wind around each other, much like a twisted ladder. These strands are made up of sugar and phosphate backbones, which form the sides of the ladder, and nitrogenous bases, which pair up in the middle to form the rungs. What's fascinating is how the bases pair specifically, adenine with thymine, and cytosine with guanine, through hydrogen bonds. This pairing is crucial for accurate copying during cell division. The helical structure not only provides stability, but also allows for the compact storage of vast amounts of genetic information within the nucleus. Understanding this structure helps us see how genetic information is maintained and passed on, which ties directly into the broader theme of how life is genetically encoded and inherited. It's really a brilliant design, elegant yet highly functional. Let's take a closer look at how DNA is organized within a cell. So, we all know that DNA is the blueprint of life, but it's not just floating around randomly. Instead, it has a very organized structure. At the most basic level, DNA strands are tightly coiled around proteins called histones, forming structures known as nucleosomes. These nucleosomes further fold and pack together to create a dense, organized structure called chromatin. When a cell is ready to divide, this chromatin condenses even more, turning into chromosomes, which are the most compact form of DNA organization. This careful arrangement is crucial because it allows the cell to efficiently manage and access its genetic information when needed. Understanding this organization helps us appreciate how genetic information is stored, protected, and precisely regulated, which ties into the bigger picture of how cells function and pass on traits. Today, I want to take a closer look at RNA structure, which is fundamental to understanding how RNA functions in the cell. RNA, or ribonucleic acid, is made up of a sequence of nucleotides, similar to DNA, but with some key differences. One of the most important aspects of RNA is its ability to fold into complex three-dimensional shapes. These structures are not just random, they are crucial for the molecule's stability and its interactions within the cell. The backbone of RNA is composed of sugar and phosphate groups, with nitrogenous bases attached to the sugars. The bases, adenine, uracil, cytosine, and guanine, pair in specific ways, forming base pairs that help stabilize the structure. Unlike DNA, RNA is usually single-stranded, which allows it to fold upon itself and form various secondary structures like hairpins, loops, and bulges. These structural features are key because they influence how RNA interacts with other molecules, such as proteins or other RNAs. The shape of an RNA molecule determines its function, whether it's acting as a messenger, a catalyst, or a regulator. Understanding RNA structure is, therefore, essential for grasping how it carries out its diverse roles in biology. In the context of this presentation, recognizing these structural characteristics helps us appreciate how RNA is not just a simple messenger, but a versatile and dynamic molecule with a rich structural landscape. Let's take a closer look at the different types and functions of the concepts we've been discussing. On this slide, you'll see the focus is on understanding the variety of types and how they serve different functions. First, it's important to recognize that there isn't just one kind, there are multiple types, each with its own unique characteristics. These types are designed to address specific needs or problems. Moving on to functions, these are essentially the roles that each type plays within a system or process. For example, some types might be primarily used for data collection, while others are geared towards analysis or decision-making. Understanding both the types and their functions helps us see the bigger picture, how these elements work together to support our overall goals. By grasping these key concepts, we can better identify which types are most suitable for particular situations and how they can be leveraged effectively. So, in summary, recognizing the different types and their functions is crucial because it allows us to choose the right tools for the right tasks, ultimately making our processes more efficient and targeted. Let's take a closer look at the bases, which are fundamental components of nucleic acids. These bases are the building blocks that carry genetic information. Moving on, the sugar component varies between DNA and RNA and plays a crucial role in their structure and function. The structural conformation of these molecules is also key, as it influences how they interact within the cell. Now, the function of DNA and RNA is different but equally essential. DNA primarily stores genetic information, while RNA is involved in protein synthesis. Finally, understanding the differences between DNA and RNA helps us see how these molecules work together in the broader context of genetic expression and cellular function. So, these points all tie back to the overall theme of understanding how genetic material is organized, stored, and utilized in living organisms. Now, let's look at some of the key functions of synthetic nucleotides. First, they can serve as antiviral agents, helping to combat viruses by interfering with their replication process. This makes them crucial in developing treatments for viral infections. Second, synthetic nucleotides are also used as anti-cancer agents. They can target rapidly dividing cancer cells by disrupting DNA synthesis, which helps to slow or stop tumor growth. Overall, these functions highlight how synthetic nucleotides are versatile tools in medicine, playing a vital role in both antiviral and anti-cancer therapies. They exemplify the broader theme of how understanding nucleotide chemistry can lead to powerful therapeutic strategies. Let's take a closer look at the central dogma of molecular biology. This concept is fundamental because it describes the flow of genetic information within a biological system. Essentially, it explains how the information stored in DNA is used to produce the proteins that are essential for life. The process starts with DNA, which serves as the blueprint. Through a process called transcription, this DNA sequence is copied into messenger RNA, or mRNA. Think of mRNA as a messenger that carries the genetic instructions from the DNA in the nucleus to the ribosomes, where proteins are made. Then, during translation, the ribosomes read the mRNA sequence and assemble the corresponding amino acids to form a protein. This flow, from DNA to RNA to protein, is the core idea behind the central dogma. It highlights the unidirectional nature of genetic information transfer, which is crucial for understanding how genes are expressed and regulated. Connecting this to our overall theme, understanding the central dogma helps us grasp how genetic information underpins all biological functions and how mutations or changes in this flow can lead to various biological outcomes. Let's take a closer look at DNA replication. This process is fundamental because it ensures that each new cell receives an exact copy of the DNA. It all begins at specific locations on the DNA molecule called origins of replication, where the double helix unwinds. This unwinding is facilitated by enzymes known as helicases, which separate the two strands to create a replication fork. As the strands separate, the cell's machinery synthesizes new complementary strands. DNA polymerase is the key enzyme here, adding nucleotides one by one to build the new strands in a precise, complementary fashion. It's important to note that replication is semi-conservative, meaning each new DNA molecule consists of one original strand and one newly synthesized strand. This high-fidelity process is crucial for maintaining genetic stability across generations. Connecting this to the overall theme, DNA replication illustrates how biological systems are designed for accuracy and continuity, ensuring the integrity of genetic information as life propagates. Let's dive into DNA transcription. This is the process where the information encoded in a DNA strand is actually used to create RNA. Think of it as copying a specific section of a recipe from a cookbook so you can follow it in the kitchen. The key point here is that transcription is selective. Only particular genes are transcribed at any given time, depending on what the cell needs. The enzyme RNA polymerase plays a central role. It binds to the DNA at a specific region called the promoter, and then unwinds the DNA strands. As it moves along the DNA, it synthesizes a complementary RNA strand, matching the DNA bases with their RNA counterparts. Remember, adenine pairs with uracil in RNA, replacing thymine. This process is crucial because RNA acts as a messenger, carrying the genetic instructions from DNA out of the nucleus and into the cytoplasm, where proteins are made. It's a fundamental step that connects the genetic blueprint stored in DNA to the functional molecules in our cells. Understanding transcription helps us see how genetic information is expressed and ultimately influences the traits and functions of an organism. Let's now talk about translation, which is a crucial aspect of our overall communication strategy. Translation isn't just about converting words from one language to another. It's about capturing the meaning, tone, and intent behind those words. When done well, it helps us reach a broader audience and build stronger connections across different cultures. One key point to remember is that effective translation requires a deep understanding of both the source and target languages, as well as cultural nuances. It's not just about literal translation, but about conveying the message in a way that resonates naturally with the new audience. This ties into our overall theme of clear, impactful communication. Whether we're translating marketing materials, technical documents, or customer support content, the goal is to maintain consistency and authenticity. By doing so, we ensure our message is understood and appreciated, regardless of language barriers. So, in essence, translation is a bridge, connecting people, ideas, and opportunities across diverse linguistic landscapes. It's a vital tool in our global strategy to foster understanding and collaboration. Let's wrap up with DNA denaturation and renaturation. Essentially, denaturation is the process where the two strands of the DNA double helix separate, typically due to heat or changes in pH. This is important because it allows us to study the DNA structure and perform techniques like PCR. Renaturation, on the other hand, is when these separated strands come back together under suitable conditions, reforming the original double helix. This process highlights the stability of DNA and its ability to reanneal after denaturation. Understanding these processes is crucial because they underpin many molecular biology techniques used in research and diagnostics today. So, in essence, these reversible steps are fundamental to manipulating and understanding genetic material.